Okay, so we're gonna go over the handouts that we did in class. We did the first one together. The first one was non-covalent interactions in biomolecules, and we did that in class. But I went ahead and posted the key to all of this, but I thought I'd make little videos of each one so that we could go through it. Okay, so when we talk about thermodynamics in living organisms, really, we want to know about the first and second laws of thermodynamics. Those are the ones that really relate to our chemical reactions. But when we think about the synthesis of biomolecules, right, that's anabolic, we're expending energy. It really kind of seems to violate our second law of thermodynamics, right? And so when we think about a biological system, the first thing we have to think about is what is it according to our definitions inside the first and second laws. So what is a system? Whoa, how about not a highlighter? What is a system? Our system is basically either you can look at it as the cell or you can look at it as the organism, right? And what are our surroundings? Our surroundings are everything else. And then together you have to remember that, okay, and that all of that together is the universe, right? Kind of talked about that in class. And so we talked about biological systems in class, and we said that those have to be really, really open systems, right? And why is that an open system? Because we exchange nutrients, waste, and then heat, energy with our surroundings. Right? And so our energy, our nutrients can be taken in by the system, and then our waste and our energy is given to the system, right? The heat that we produce is actually given to the surroundings. So what are those first and second laws, right? And why did we think we're going to be violating them? So what's our first law? Our first law is that energy is not created or destroyed. B-I-S-D-E-S. Oh, if I could spell. But they can be converted, which is really important to remember. You can convert it. You can interconvert it. And the second law tells us that all spontaneous processes in our universe tends towards the dispersal of energy, or we're going to see an increase in disorder in the universe, right? So how is it that we can build these big macromolecules and all those anabolic pathways when all of our universe is supposed to be tending towards disorder, right? So how can cells still follow both the first and second laws of thermodynamics? That's a really interesting question that you really have to sort of think about. And I want you to try to answer this before I do. So pause it, try to answer it before I do. So if you think about it, right, in the first law of thermodynamics, our biological systems are open, right? So our biological systems are open because you're obtaining energy from the surroundings, right? You're eating, and you're going to convert that to chemical energy. So they're open. So we get energy from surroundings and we convert it. Chemical energy. I'm afraid if I don't zoom in, I'm never going to fit this in. Okay, and that chemical energy can now be used inside the system. So the chemical energy used by the system. So inside the system, you have to think that we are converting. So think about plants, right? Plants convert solar energy into chemical energy. We, on the other hand, convert chemical energy into another kind of chemical energy. But regardless of whichever one you're thinking about, we're using that energy to synthesize molecules that are more complex than their precursor molecules. And that really seems to violate the second law of thermodynamics, right? So our precursor is sort of complex, right? That hamburger that you ate had carbohydrates and protein and all that kind of stuff in it. But then you're going to make a whole cell and all the components inside of the cell. So you're trying to make molecules that are more, right, complex. And by more complex, that would be a decrease in entropy than our precursor. And this is what is our violation. And I'll put it in quotation marks because it's not really a violation, but it seems like energy isn't being dispersed, right? But what you have to remember is that biological systems are using other processes that degrade biomolecules into smaller, chemically simple biomolecules, right? So at the same time, you have inside the system, so the system, the cell, the organism, is using other pathways, other biological processes that degrade biomolecules. Let me see. Into, so sad you'll have to like look at me spell, into smaller, chemically more simple molecules, right? And so that's actually a dispersion of energy. So here, this second thing is a dispersal of energy, right? So the energy is being expelled into the surroundings, right? Because we're going from a very complex thing that we just ate into breaking it down into individual amino acids, right? So we're actually seeing an increase in entropy. So this would be an increase in entropy. And so there's a balance between synthesis and degradation. I'm gonna have to like take up all this room. So we find a balance between synthesis and degradation. And that allows the cells to convert energy into different forms while still obeying the first law, right? So because you can't create, you can't destroy, but you can interconvert. So if we have a balance between synthesis and degradation, then we're allowing the cells to convert energy into different forms, but still obey the first law of thermodynamics. Then we can exchange the degraded molecules with the surroundings, and then that's going to increase the entropy of the universe in general. So we'll have waste products eventually that are small, and they're going to be expelled out into the surroundings. And in the end, the waste products that we make, so waste products shared with our surroundings, are going to be very, very small and degraded, are small and degraded. And so that is going to overall we'll get an increase in entropy of the universe. Because we're not just looking at what goes on inside the cell. We have to look at not only the cell, but also the surroundings. And so we really are obeying both the first and second laws, even though it really seems counterintuitive of our second law that we're tending towards disorder, but it's really disorder of the entire universe. So you have to remember that we're talking about the universe, not just what's inside the cell or what's inside the organism, right? Okay. So then I want to talk about buffers and Henderson-Hasselbalch, right? So we did quite a bit of this in class, but sometimes it's nice to have kind of a refresher, right? So this is should be mostly a refresher of what you've done in intro chemistry. So would a strong acid or a strong base be able to act as a buffer? And you have to remember that buffer that we had inside of the lecture, right? So we had something that had a hydrogen attached to it, and we just make that as HA, right? And we say, okay, if it's weak, right, then we can go and we can make the conjugate, but it can also plus H plus, but it can also go in the reverse direction. And that would be something that is weak, but something that is strong is HA, and we have a single arrow, and we're going to make A minus plus H plus. You can't go in the reverse direction. You can only go in the forward direction for a strong. This is a strong, Okay, so it's kind of just what you have to remember with that. So would a strong acid or base be able to act as a buffer? And the answer to that is no. No, absolutely not. So a buffer, what's the point of a buffer? A buffer has to be able to convert a strong acid, and I always use these abbreviations. So it has to be able to convert a strong acid into a weak acid, but at the same time, it has to be able to take a strong base and convert that into a weak base, right? So it has to be able to have a buffering capacity for strong acids or strong bases that are added to the solution. So then by definition, what is a buffer? Well, a buffer must be a weak acid and its conjugate base, or a weak base and its conjugate acid. Either one is okay, right? So if we look here, here's a reaction, right? We have a couple of different things going on, and we want to first identify the weak base and the conjugate acid, and then we're going to look at what happens when we actually try to buffer this solution, right? Okay, so let's kind of zoom in. So H2CO3, we can lose the hydrogen. So this is going to be our weak acid, right? And then if you look, where's my conjugate base? Well, the conjugate is all I'm looking for is the same formula, but one less hydrogen. So here I can see that I've lost a hydrogen, gained a negative charge. So this is my conjugate base, right? Okay, so now what happens if we take this system? We have a weak acid and we have the conjugate base, and we have them both because we have this double-sided arrow. So what happens if I add something that is a base to my buffer? Well, what part of the buffer is actually going to interact with the base? And it's going to be the opposite. If this is a strong base, I want my weak acid to react with the strong base. So my weak acid was H2CO3, right? So what's going to happen is we're going to have a reversible reaction where we're going to make the conjugate base, which is HCO3 minus plus water, right? So we just turned a strong base. Guess what? This is the conjugate base, but it's also weak. It's a weak base. We were able to turn a strong base into a weak base, and that's what we really want from a buffer. Okay, so now that's an example. An example of that would be if I added sodium hydroxide we talked about in class, because if you put sodium hydroxide in water, what happens? It 100% disassociates into Na plus and OH minus, right? So that's where the OH came from. So now what if I do something else? What if I do, if I add HCl? Well, HCl is an example of a strong acid. So we know in water, it will completely disassociate into H plus Cl minus. So here's my H plus. That's what I'm worried about, right? This is my biological system. I can't have big changes in pH. So adding H plus is really dangerous. So I'm not going to represent that as H plus anymore. I'm going to actually represent that as H3O plus, right? And to that, I'm going to add some part of my buffer system that is going to be able to soak up those extra hydrogen ions. And so look in your equation above, what part of the buffer? Do I want the weak acid or do I want the conjugate base? Well, in this case, I want the conjugate base. The conjugate base is HCO3 minus. All right, so I know that this is a buffer, it's weak. So we're gonna have to have our double-sided arrow. So what's gonna happen here? Well, if this is our weak base over here, what's going to happen when it interacts with a hydrogen ion? We're going to make a conjugate acid. So H2CO3 plus, we're going to have leftover water, right? Because this one hydrogen just gets picked up by our weak base. So I don't think I labeled this. So this is the weak base, and this is the conjugate acid, right? So what we've done is we've basically taken a strong acid and we've turned it into a weak acid. And that's how buffers work. That's how buffers handle big introductions of acids or bases into a biological system, right? We get rid of the strong part and we turn it into weak, which is something that we can handle. Okay, so Henderson-Hasselbalch is derived from which equation and why is it useful? Okay, so let's kind of talk about that. So remember, when we talk about disassociation of an acid, a weak acid, so we have HA and we have it disassociating reversibly to H plus our conjugate base, A minus. So from that, we can write a Ka. We can write, this is what we're kind of derived from, our equilibrium expression for our acid. So we write that's H plus A minus over HA, right? Reactants over, products over reactants, excuse me. If I do this and I solve for this guy right here. So if I solve for H plus, what do I get? Well, go back to my other color. I want to isolate concentration H plus. So I should get Ka is equal to, and I'm going to flip flop, and I'm going to get HA over A minus, right? Okay. So now if I take that and I take the negative log of both. So I'm going to do, let me write what I'm kind of doing. So from here to here, I'm taking the negative log, right? Because Henderson-Hasselbalch, remember, has that pKa, right? So where did that pK from? It came from doing the negative log. So that's what I'm going to do, that equation that was down there. So I'm going to take the negative log of Ka times HA, A minus, right? Close parentheses, equal, I have to do it to the both sides, negative log of H plus. Oh, what is negative log of H plus? Oh, that's pH, right? Okay, so now you have to distribute within here. So you get the negative log of Ka, and then that is going to be the plus the log of HA, right? To the minus 1 over A to the minus 1. Right? So you can make it a positive log if it's raised to the negative 1. So all we have to do is invert HA and A. And you don't have to be able to do this derivation. You don't have to be able to do this. It's just, it's important to understand where it comes from, but you don't have to be able to replicate this. So I get negative log of Ka is just pKa, right? So plus, I'm sorry. I did this as, this is supposed to be negative. I jumped ahead of myself. So if I have a negative log and then I raise these to the negative 1, right? So here's what it originally is, I'm sorry. And then if you do to the negative 1, let me see, let me change colors. So then if I do this, do it to the negative 1, that turns that into a positive. I just kind of did it, sorry. So now I'm gonna bring it down. So I get the positive log, positive log, and if it's raised to the negative 1, all I have to do is flip-flop my numerator and denominator. So I get A minus over HA, right, is equal to pH. Well, this is the Henderson-Hasselbalch equation, right? So basically, we just got it from the equilibrium expression of the acid. That's all we did, right? And so why is this useful? Why do we want the Henderson-Hasselbalch equation? Because it relates pH to pKa, right? So you just have to remember that it's a way for us to look at what kind of buffer we want for a particular system, right? Because we know when using the Henderson-Hasselbalch equation, if we have pH is equal to pKa, that is going to be the buffering point of our buffer. So our pKa will tell us, I want a buffer that's going to pH, that is going to buffer a pH 8 solution, right? So then I need to go and find a buffer that has a pKa that's pH 8. Well, what if I, in another case, want to have a buffer that's going to buffer at pH 5? Well, I need to go and get a different buffer because I need to get a different pKa, because a buffer is only good for plus or minus 1 pH value from the pKa. So each buffer has a unique pKa value, right? Because it has a unique dissociation constant. So therefore, you need to find the right buffer for the right system. Okay. All right. Number 4, right? If a buffer is prepared with equal amounts of weak acid and conjugate base, how is the pH related to the pKa? If the buffer is made so that the amount of weak acid is greater than the amount of conjugate base, what will be the impact on the pH? Oh, I'm gonna have to make a second recording. Okay, I'll make a second one. Sorry, y'all.